So, dear colleagues, let me start our seminar. This is the first seminar in this year. Therefore, Merry Christmas and Happy New Year. I hope we will victory during this new year, 2023. And today our lecturer, Dr. William Scott, who is a research professor in the Department of Chemistry and Chemical Biology, Indiana University, Purdue University, Indianapolis, Indiana, USA. He got his bachelor's degrees at William College and doctorate at the University of California, Los Angeles. Following postdoctoral work at Rockefeller University and Caltech, he joined Eli Lilly and worked for 27 years in drug discovery and the development of new research technologies. He holds patents in the areas of enzymes inhibition, anti-cancer agents, Alzheimer's treatments, monoclonal drug conjugates, protein drug linkers, antibacterials, solid phase synthesis equipment, and amide coupling reagents. While Eli Lilly, Dr. Scott taught part-time at Butler University and Purdue University. In 2002, he moved to a research professor position at the Purdue University, where he currently teaches, conducts researches, and publishes on a wide variety of topics. He has special interest in the educating global students in the drug discovery process and the skills required while they participate in the research to find drugs for diseases not being addressed by the pharmaceutical industry. To do this, he created the Distributed Drug Discovery Program. It involves chemistry students and multiple institutions throughout the world in the distributed problem-solving process, and has been implemented at cities in Spain, the Czech Republic, Cuba, Mexico, Puerto Rico, Poland, along with 6 academic institutions in the continental United States. He has presented lectures on distributed drug discovery in Barcelona, Newcastle, Oxford, Genoa, Basel, Prague, Krakow, Lublin, and today here in Kharkiv Chemical Seminar. So, dear Bill, please.
Thank you. Thank you very much, Professor Chebanov. I must say it's an honor for me to be invited to give a seminar here. I think you've put together a very impressive program, and I hope that this seminar will be another sign to everybody listening that we support you, and we support you both academically, but we also support you in terms of what we can do in the United States to supply the appropriate equipment to fight this battle. And I'm looking forward to the time when this is all over and I can visit people that I've met through Zoom here in Ukraine, and we can work together under peaceful conditions. So I'm going to tell you about a program that I call Distributed Drug Discovery. And I'll first give you a little background on how this project originated. It started out, let me see if I can, I left Lilly in 2003, and within a few months of when I left, there was this tragic re-entry of the shuttle from outer space, in which one of the tiles, some of the tiles had come loose, and because of the friction and the heat generated, the shuttle disintegrated on re-entry. The day after that, this was a picture on the front page of our paper, and while it represented a tragic event, at the same time, what occurred to me was it was wonderful that a challenge to try to explore outer space had brought together people from many different backgrounds. And so I started thinking about how, if we have a really powerful goal and it's something that involves multiple disciplines, they can be brought together in a very concentrated way to try to solve that problem. So this would be the case with exploration of outer space. Obviously, you need all these kinds of different expertises to carry it out. Well, I just come from Lilly, and in that case, we also had a powerful challenge, and that was to discover drugs. And what I loved about working at Lilly was, unlike in academia, where many of the disciplines are siloed and people focus only on chemistry or biology or computational analysis and don't have the incentive to work together to solve a common problem, unlike the situation in academia, in the pharmaceutical industry, they absolutely had to have these people cooperate with one another to solve the problem. So I just give an example, some of the disciplines and areas of expertise that are needed. However, one of the things that is an unfortunate side effect of pharmaceutical research is there is another really key piece of that, and that's a profit incentive. Now, I've got to look at that from my career at Lilly as a positive because I got paid quite well. But on the other hand, what it means is that the pharmaceutical industry picks drug discovery challenges which are going to make them a profit. And so there are very, very important diseases out there that aren't being addressed with the same focus as these diseases like cancer or central nervous system diseases or hypertension that are the focus of pharmaceutical industry research. So I'd like to, I was challenged really to try to figure out a way, now that I was back in academia, that I could use the skills and understanding that I'd learned at Lilly, but apply them to some of these diseases that aren't being commonly addressed by pharmaceutical companies. And I just give two examples here. One is of a child has leishmaniasis, not an issue in the United States, but obviously an issue in many countries throughout the world, especially, for example, Afghanistan. And that represents a large population of people, but they don't have the money to pay for the drugs that might be developed by a pharmaceutical company. So the profit incentive is not there present for looking at ways to treat that. Another category are commonly called orphan diseases. These may involve people anywhere in the world. They may be in societies that have relative wealth, but the population of patients in these areas is so small that, again, there isn't the same financial incentive for a traditional pharmaceutical project to find drugs to treat these diseases. So with that in mind, having come from the pharmaceutical industry, wanting to see if we can make some impact on some of the diseases that aren't being addressed by the pharmaceutical industry, I came up with a program which I call Distributed Drug Discovery. And now we're going to put at the core of our challenge, neglected and infectious disease drug discovery. And we're going to bring in some things that aren't normally a focus in the pharmaceutical industry, education and humanitarian concerns. So that was the driving force behind coming up with this program, Distributed Drug Discovery. Obviously, we don't have the resources that we would have at a traditional pharmaceutical company like Merck or Pfizer or Lilly or Genentech or a lot of these other companies. So we had to think of a creative way to combine both education and skills that are significant enough to make molecules that could have an impact in either neglected or infectious disease. So here is the bottom line scheme that I came up with back in 2003, and I called Distributed Drug Discovery. And the idea was, if we could find some way of converting well-known procedures in the literature, sometimes pretty sophisticated, sometimes pretty complicated, but are powerful in that they can make molecules that could be potential drugs. If we can convert those procedures into procedures that enable students anywhere in the world to make molecules, and we generate computationally the molecules that will be accessible by these procedures into a large virtual catalog, do some computational analysis or expert analysis to whittle down this large virtual catalog into a smaller collection of molecules. It's smaller, but it still might be large. These virtual catalogs really, as you know from Enamine's experience, can be on the order of millions of compounds. Let's say we whittle that down to a selection of maybe 200 molecules that look like they would be good candidates for a proposed neglected disease or infectious disease target. Here's where education comes in and the distributed concept comes in. What if instead of having a facility like Lilly with a whole bunch of chemists there working, making those molecules, what if we take that set of 200 molecules, and if the procedures are simple enough, if the equipment's inexpensive enough, we can have those molecules made at distributed sites at schools throughout the world, where instead of one school making 200 molecules, they can make 5. Another school with more resources might make 20 or a larger number of students. Another school might just make 6. But when you put together a large collection of schools that have taken a subset of these molecules that we propose as potential treatments. They could be synthesized, again, if we have simple equipment, powerful equipment, reproducible procedures that students can do. They could be made in these distributed sites and then combined into this collection of molecules now available for screening. If we use an analogous approach in the screening area, what if we have biology laboratories that are not only focusing, I'll take bacteria, for example, as we want to find the drug to kill a particular type of bacteria. Well, in a particular biology lab, a microbiology lab, they would be looking at the growth of bacteria. Well, why not have them grow that bacteria that is a disease target? And while they're examining and learning about how that happens, they take the molecules that the students across the hallway have made in their laboratory and test them in the biology laboratory. So they're not only doing theoretical work on material that's already known, but they're doing exploratory work to try to determine if new molecules made by the students can now effectively inhibit that bacteria. And I'll give you an example of that in a few minutes. So that's the idea. So let me just kind of break this down into the individual components of distributed drug discovery. Before we involve students at the distributed chemical synthesis stage, we have to be able to carry out the stage one, where we have converted well-known procedures from the literature in the D3 procedures that are simple, inexpensive, powerful, and then show that students can carry them out and then create these large virtual catalogs. So here are the steps that we go through. So we're going to go from D3 directed basic research to virtual catalogs. The first step we do is to go through the literature and see what particular procedures already published might be appropriate to convert into student procedures that are compatible with laboratories that they would be conducting either as undergraduates or graduate students. When we've done that, here at IUPUI, what we do is we have an independent group of undergraduate students who we work with to examine these procedures and work with them to convert those procedures into things that are so readable and well documented in a written up procedure that students will be able to carry these out in a regular undergraduate laboratory. So that's the first part of our process, is to convert some not theoretical, but published procedure into what we'll call a D3 enabled synthetic procedure. And this is an example of the first procedure that we did in distributed drug discovery. My colleague, Dr. Marty O'Donnell, and I had published back in the Journal of American Chemical Society a while ago, 1996, a procedure where we have a resin-bound precursor to an unnatural amine, and we can convert this into this key molecule here. All this is an unnatural amino acid bound to polymer beads, and I'll talk a little bit more about the power of polymer beads down the line. But the important thing was that we had a nice procedure to expand the set of amino acids from the traditional 20 natural amino acids bound to resin that you can purchase for peptide synthesis to hundreds of different unnatural amino acids, racemic, so we get both enantiomers here. And these then served as starting material for a lab where we then just simply acylated with a carboxylic acid here, the unnatural amino acid, and then cleaved it from the resin, and we get this molecule. Now that looks not all that complicated, but in fact, when you see the kinds of structures that you can make from that, they can be pretty sophisticated. But this was a lab that our students converted into a D3 lab that could be reproducibly carried out by students using simple equipment. So that's the procedure. Here's the simple equipment. Every single step of the synthesis that I just showed you can be carried out with this equipment. You don't need you don't need separatory funnels. You don't need fancy equipment to do chromatography. You don't do recrystallizations. Everything is done in this reaction vessel. And courtesy of solid phase synthesis, you can go through these steps, these intermediate steps, by simply filtering off all the reagents and byproducts and then getting ready for the next step. So this is all they need to do that chemistry. This just shows you a picture of some of our students at IUPUI carrying out the chemistry. Nothing really sophisticated, but I would like to just show you our driving force for inexpensive. So we don't have always access to lab jacks. Certainly we do at IUPUI, but if we're doing this in Cuba, say, they might have a limited number of lab jacks, or Puerto Rico. And so we just hammer together two, we call them 2x4s, pieces of lumber, to make the appropriate height to put this drain tray where they've got these reaction vessels that are draining out the liquid that was containing the reagents. The beads are kept from going through by a filter, and then it drains out into the speaker. So that's our expensive equipment, 2x4s nailed together. But that's a driving force again in this project. We need to make it simple. We need to make it inexpensive, so not only IUPUI can carry it out, but other schools throughout the world can carry it out. So once we have the D3 enabled chemistry, we then go to the process of creating these virtual catalogs. And so we first search for compatible reagents with that synthetic sequence to come up with a set of compatible available reagents. And then this just reminds you of the synthetic procedure that I outlined, but we'll have two variable steps in the synthesis. One is R1X. X is a good leaving group for this. It's actually an alkylation type process. So we introduce R1 with R1X's, and then R2 is introduced as a carboxylic acid. So we go back into the literature and to supply houses to find out what alkylating agents and what carboxylic acids are available commercially, and use our own common sense or data from the literature to identify those that we are comfortable with, expect that they will be good reactants and work in this procedure. And this just shows you a small collection of the alkylating agents that we identified from this particular search. All of them, most of them being halides, are being displaced, but there are also electrophiles here, like Michael addition type electrophiles. Then here are another limited set of carboxylic acids. So through this search, we identified 100 different R1Xs and 100 different carboxylic acids. And now we were ready to go through the next stage, which is to convert them via a computational enumeration, using that procedure that we've documented, into a large virtual catalog. This will be, in enamine standards, not a very large virtual catalog, but it illustrates the concept and the process. So we have 100 R1s that were available that we selected. We used some other tools to limit those to make sure that they were different enough in structure that they represented unique kinds of categories of molecules. So we did some computational processing there. But from 100 R1s and 100 RXs, you know, from a combinatorial point of view, that for each one of these R1s, you can attach 100 R2s. And since you have 100 R1s, you got 100 times 100. So that gives you 10,000 possible combinations of just 100 R1s and 100 R2s. The reason we put up here 20,000 is the chemistry that we use generates racemic molecules. So it's a mixture of the two enantiomers. So for every one of these molecules, we get two. So we get 2 times 10,000 or 20,000 different molecules. I worked under Dave Evans when I got my degree. Dave Evans, maybe some of you know, is really an expert in chiral synthesis. Dave would smile when I go back and make these things racemic. But the point here is, in fact, we desire racemic molecules because we're only at the very early stages of drug discovery. And in many of the cases, we would like to have both enantiomers present, so they would see if one of the two had biological activity. And then we have the tools later on to go back and synthesize the individual enantiomers or separate them via chiral chromatography. So that's the enumeration process. We used sometimes ChemAxon software, which I think some people are familiar with in Ukraine. I believe that that's a software that's used by enamine, although I'm not sure of that for constructing their virtual catalogs. So here, now with some flesh on those molecules, shows you that what appeared to be a simple unnatural amino acid with the amino group here, the carboxylic acid, an R1 here, and an R2 here, you get some pretty interesting molecules that are all accessible by undergraduates using this procedure and this simple equipment if they have the reagents available. So that's how we go about the process of generating these virtual catalogs. And we now have a whole bunch of D3 procedures to generate other virtual catalogs, and we're in the process of getting support to enumerate those catalogs and make them available to everybody to search. So now, and I won't talk about the details of doing computational analysis, that's a whole other seminar, but let's say we've taken that virtual catalog and we've identified the proposed drug leads. How are we going to go about actually making these in a distributed fashion? So I'm going to focus now on this particular stage of the distributed process, the distributed synthesis of these molecules. And I just show you some pictures, snapshots of pictures from workshops that have been done either IUPUI or throughout the world. So this is, we have in Lublin, Poland. I mentioned to Valentine earlier, my previous wife who passed away grew up in Lublin, Poland. So I have an affection for Poland, and we did this project in Lublin. But we've also done it in Cuba, we've done it in Mexico. And these are just some of the snapshots. We've done it in Barcelona, as I mentioned before. We've done it in Puerto Rico. So students love it. They like the idea that they're not just making molecules that are going to be thrown in a trash can or a disposal facility at the end of their synthesis, but they're making molecules that are going to be tested and could be potential drugs to treat these diseases. By the way, it's a great way to bring people together. So we, and when we went to Cuba, we brought down a couple of our students as TAs, and it was just wonderful to see the interaction of our TAs with these students, where we could learn about each other in a non-threatening way. Again, focused on solving problems we could all agree are problems that are appropriate to solve. So let me give you an example of one of the projects that's ended up distributed. And this began with an observation, well, actually a report in the literature, subsequently actually not repeated, but this was, the report was that D-tyrosine, this very simple natural amino acid in the other configuration than the naturally occurring configuration, was able to influence the formation of biofilm by Pseudomonas aeruginosa. This was where we linked back into an orphan disease. We had a colleague in the biology department here at IUPUI who was very interested in cystic fibrosis, so we could team up with him. We know that Pseudomonas aeruginosa is a very nasty bacteria for people who have cystic fibrosis. So this put us in the realm of looking for treatment for cystic fibrosis by inhibiting the growth of Pseudomonas aeruginosa, inhibiting the formation of biofilms. So we looked at that molecule, and right away, you know, from just that first D3 lab that I showed you, we have ways of making racemic unnatural amino acids on resin and then chopping them off the resin and getting these unnatural amino acids. So we decided to have a project where we made analogs of tyrosine and phenylalanine, where there'd be all sorts of different R groups attached to the phenyl group, accessible by a student-run D3 procedure, and test them against Pseudomonas aeruginosa. So the first lab that we developed to do this was very, very simple. It did that alkylation chemistry that I showed you earlier with a set of benzyl halides, where now we had different R substituents. And we limited it at first to just different halogen atoms. So there's either fluorine, R was either fluorine, a chlorine, a bromine, or an iodine. And they were at either the 2, the 3, or the 4 position on the aromatic ring. And sometimes we had multiple halogens in the aromatic ring. And once that reaction had been done on resin, we could cleave it, the product from the resin. We didn't acylate it like we did in the initial D3 lab. We just took the first variable, the unnatural amino acid, and cleaved it from the resin. And we got 20 unnatural tyrosine or phenylalanine analogs. Well, so this is the chemistry component. We need to test these molecules. So at the same time that we're doing the chemical synthesis, we're working with our biological colleagues to develop a D3 biology lab that would test them against Pseudomonas aeruginosa. And here's the simple assay. I love things that are simple and visual. And so we have here just a simple plastic well, 24 well tray, typical of biology laboratories. And in it, we put the Pseudomonas aeruginosa in every one of these wells, except the last 2 wells here. And so we put the Pseudomonas aeruginosa in all those wells. Then we would come down, it being a particular media. We do, because we are strong believers in replication. This is really key to our program. It has to be replicable by multiple students. In this case, this student in their own hands would replicate the work. So across row A would be the same set of compounds that went across row B. So we'd take 6 different molecules made by the students in the lab, and one of them would go down column 1, 2, 3, 4, 5, and 6. So we test 6 molecules in replicate in these 2 rows. And then we'd have controls in these various wells here. We'd let the bacteria grow. We'd, after 24 hours, 37 degrees, we'd suck out the bacteria. If the Pseudomonas aeruginosa grew, it will often form a biofilm, and the biofilm sticks to the plastic. After we've removed the bacteria, we can wash away any residual bacteria, and then we can stain the bacteria with this crystal violet. And when we add acetic acid after we've stained it, we get this nice, pretty color. For those wells in which the Pseudomonas aeruginosa has grown, formed biofilm, and it got stained. So this tells us that whatever compound that we put in column 2, 3, 4, 5, and 6 at that particular concentration had no effect on the growth of Pseudomonas aeruginosa and forming biofilms. We had the controls here where just bacteria alone shows, yeah, we form biofilm. There's nothing in there to inhibit the growth. We had other things that should inhibit the growth, and those were then controls showing that the experiment was working. And then we looked specifically column 1 and said, boy, that looks interesting. That whatever compound the students made in the chemistry lab, that one is able to inhibit the formation of biofilm by Pseudomonas aeruginosa. So now we had 20 different unnatural amino acids, racemic amino acids. We had a biology lab, so we put the 2 together, and out of that came, by the way, we had over like 100 biology students in multiple lab sections doing this experiment. So you can imagine the degree of replication that's going on here, because some of those plates would be identical. They'd have the identical compounds, but another student would be doing it. So it's a powerful illustration to the students of a fundamental value in science, which is reproducibility. So out of that came reproducibly these 4 molecules that inhibited the growth of biofilm at 1 to 5 micrograms per mil. Now, this is actually pretty significant because that's a pretty low concentration. It turns out that tobramycin, which is used to treat Pseudomonas aeruginosa infections, its activity is shown at about the same level. So these were pretty potent at inhibiting the growth of the bacteria, just simple fluorinated phenylalanines. Well, as you might expect, after we saw this, it was time to go into the literature and see what was known about fluorinated phenylalanines. And it turned out there was a lot known about fluorinated phenylalanines. They'd been made as early as the 1940s. And there was a report fairly early on that the parafluorophenylalanine was a potent inhibitor of Pseudomonas aeruginosa growth in bacteria. So our students, unbeknownst to us, we rediscovered this class of molecules. So nothing new here in terms of what we did, although the process was affirmed that we could do this without having any understanding prior that the fluorinated phenylalanines would have that activity. As we went into the literature deeper, it was also clear that these fluorinated phenylalanines not only inhibited bacterial growth, but they were quite toxic to mammals. So they're not going to be a drug. So this is an opportunity to go back and teach our students all the time. We're in this process. We're also telling them about the drug discovery process. So we had it really simple early on, but when we get to this stage, we let them know drug discovery is a very complicated process. And so it may be one thing to get a molecule that in a cell-based assay is showing activity and you've made it, but you have a long ways to go before you improve drug. And it was this part here that I then bring up to the students, toxicology. So we've got to make sure that whatever molecule we have that looks good to treat a disease is not going to be toxic to the person that we're treating them with. And so that ended up taking us to our first distributed, now outside of IUPUI, drug discovery project. And that was to find drugs that treat cystic fibrosis based on this initial discovery or rediscovery of the fluorinated phenylalanines. So what we did was we said, and this is from my experience at Lilly and the literature, knowing that a lot of times you could take a drug molecule, or not a lot of times, but sometimes you could take a drug molecule that has a lot of promise, but drawbacks at the same time, and convert it temporarily into a new molecule that disguises the active molecule within it and gives you the desirable properties until it's time to reveal that molecule in its active form. And those are called prodrugs. So here's an example of a dipeptide prodrug that we envision being a possible solution to the toxicity associated with the parent fluorinated phenylalanine. We were going to make a whole series of dipeptide prodrugs here with the thought that we already knew that Pseudomonas aeruginosa has specific dipeptide transporters to take dipeptides into the cell. So maybe we can selectively get a dipeptide that isn't taken into mammalian cells, but is selectively taken into Pseudomonas aeruginosa by virtue of these transporters. Once in there, we also have the possibility that there'll be a bacteria-specific peptidase, a peptidase that we don't have, but that the bacteria has, that is able to recognize this dipeptide and cleave it back into the constituent fluorinated phenylalanine plus whatever other amino acid we had hung on the other side. The bacteria, much to its surprise, has now, in its search for nutrients, using its peptidase to degrade naturally occurring dipeptides, has taken this unnatural prodrug dipeptide, cleaved it back into the fluorinated phenylalanine that's going to kill it. So that was the idea. And so we decided to make a distributed prodrug project based on that. And here's the simple synthesis that our undergraduates then in our independent group worked out, and then we showed could be done at IUPUI, and then we took to a number of different schools in the United States and elsewhere. Very simple. This is basic peptide chemistry. The only difference is that we're using an unnatural amino acid to couple to natural resin-bound natural amino acids. So here you see the combinatorial possibilities and the challenge if we're going to just have a single chemist making all these things. If we have 20 natural amino acids, each one of those could be combined with any of the 4 different fluorinated phenylalanine. So we'd have 20 times 4, 80 different possible molecules to explore in our biology for the potential that they are active against Pseudomonas aeruginosa, but not toxic to us. So here's the grid that we computationally construct. And what you'll see here is we organize it by either the 2-fluorophenylalanine at the N-terminal position, the 3-fluorophenylalanine at the N-terminal, the 4-fluorophenylalanine at the N-terminal, or the 3,4-difluoro. And then each of the 20 different amino acids are on the other side of the molecule. So that was a challenge. So we then talked to our collaborators at Santa Clara University, Colorado College, Goshen College, and a local university about whether or not they'd be willing to help us make subsets of these molecules. And they did. And this is the current state of this project, where in blue, we've made 61 of the total 80 possible molecules. Some of the ones that you see in red are going to be more problematic. We may have to have not a regular undergraduate in a normal undergraduate lab make them, but do a little bit more high level work. The thiol group on cysteine makes it somewhat problematic in terms of oxidation and dimerization, things that go with that. But 61 and counting. So we're still working on this project. And here are some of the schools that have been involved. Again, I mentioned these, and we've done pilot scale dipeptide prodrug work in Mexico, University of Indianapolis and Puerto Rico, and also courtesy of a very wonderful colleague, Daniel Garcia Rivera at the University of Havana. Well, we were filling in some of the blanks on this project back in 2020. And things turned around quite rapidly. So we had a lab going, but we had to shut the lab down in March of 2020 when this new challenge came up. It's not something that's not being addressed by the pharmaceutical industry. It certainly is, but we saw an opportunity to bring in distributed drug discovery here too. So obviously we all know about COVID. So we shut down the lab and immediately worked on developing a distributed synthesis of potential drugs to treat COVID-19. So I'll use the remainder of the seminar to tell you how we approach this and where we stand right now, and also to invite you to participate in it. So just a really simple schematic on how SARS-CoV-2 replicates. You're all quite aware of the binding of the virus to the cell surface via spike proteins. You'd have to have your head in the sand not to have heard about all the vaccine work that's been developed to try to get antibodies to the spike protein. So that if you get antibodies to the spike protein, you can competitively bind to the spike protein and prevent the binding to one of the receptors on the cell surface that's going to allow the virus to get in. Those antibodies can come naturally from us getting infected. They can come from a messenger RNA-based vaccine. You can actually produce the antibodies commercially. Some companies do that and then infuse them. In any case, once the virus gets inside the cell, the key piece that we were going to focus on is the RNA that is released by the virus codes for the synthesis of really large polyproteins. And those polyproteins in and of themselves are not going to function in replication capacity for the virus. They need to be proteolyzed. They need to be cleaved into individual proteins that are contained within that polyprotein. So the polyprotein has multiple proteins stitched together in this large polyprotein, and an enzyme called main protease, or MPRO, or 3CL sometimes, is the key protease present in the virus that cleaves out the individual proteins from this large polyprotein. So it's pretty clear to everybody in this area that if you can get a good inhibitor of that protease, you've stood a chance of getting a molecule that could inhibit virus replication. So that became a target, obviously not just for us, for many people. So I'm going to tell you the thought process that goes through designing a drug to inhibit this MPRO, this main protease, and then how that ended up resulting in a drug that's used to treat COVID. In fact, I took the drug. So I love being able to be hands-on sometimes. And in this case, I actually got COVID and I was able to take a drug that came out of this approach. More on that later. So here's the polyprotein, but I'm showing it schematically with embedded within it protein A, B, C, and D. The key here, though, is that in the polyprotein, there's an amino acid sequence right before the bond that's going to be cleaved by this main protease that is almost universally a leucine and a glutamine. The glutamine is universal. The leucine is almost universal. Most of the times in the sequence that's going to be cleaved, it'll be a leucine followed by a glutamine residue. There's a pocket in the enzyme that recognizes the leucine and glutamine, pulls it into the enzyme's active site, and within the active site of the enzyme is a cysteine that contains an SH group. That SH group does the simple, chemical reaction. It attacks the carbonyl, forms an intermediate that falls apart, and it's that thiol that catalyzes the cleavage of this bond to generate protein A, protein B when the bond is cleaved over here, protein C cleaved over here, etc. So the idea is, and was followed by researchers effectively to come up with a drug to treat COVID, is what if we look at that sequence, the conserved residues, leucine and glutamine. We don't want to affect other proteolytic processes that are important for us. So if you can find the sequence that's unique to the virus and an enzyme that's unique to the virus and inhibit that, then you've got a much better shot at having some a safe biologically active compound. So these are the residues, and I'm going to use an untraditional nomenclature or way of depicting this. I'd like to think of, as I'm not actually a fisherman, but I've observed fishermen. I'd like to think of this portion here as being the bait. And that's what's going to be recognized by the active site of the enzyme. So the bait portion is something we're going to want to preserve in a potential drug. And once we've got the bait portion there, we're going to want to make it an even tighter situation. And in my simplistic way of thinking, the bait could be a lure of some sort that imitates that sequence here. and is closely enough resembling it that the enzyme takes it into its active site. Once it's in the active site, we're going to put a hook on. And in this case, the hook in our fishing expedition is going to be a hook that binds quite tightly to the thiol group. This is on a more sophisticated level, the kind of reasoning that went into the researchers that started looking at inhibitors of MPRO as potential ways to treat COVID-19. And that same year, 2020, after we started thinking about this project, there was a report in Science of a molecule that exemplifies this concept. Here, the bait, you can see what they've done here is they've conformationally restricted the glutamine residue, and they have the leucine residue tied back into a 6-membered ring. You might ask why they didn't just have leucine and glutamine. Shoot, that's the actual good bait for the enzyme. Well, it's one thing to have something that'll bind to the enzyme. It's another thing to have it as a drug. So there are going to be other issues with glutamine and leucine in terms of the availability of that molecule, the stability of that molecule, the ability to make it, all sorts of other issues. And so people didn't use explicitly leucine or glutamine, but in fact disguised it already right from the start in these lures for the enzyme. And now their hook was going to be an aldehyde. And you can see here just schematically, aldehydes will react with thiol groups reversibly, but pretty tightly to form this intermediate here. And so they made that looked pretty good as an inhibitor of MPRO, but it wasn't something that they were taking forward at that time to go through that elaborate process to come up with an actual drug. In December of 2021, that's barely a year ago. So a year ago, December, there was another report in Science that really changed the area. And that was a report that this molecule here was able to very potently inhibit the enzyme. And you see here, in fact, they did keep the leucine. They have other groups out here to explore other parts of the pocket that aren't part of the naturally conserved area. But everybody in this area must be the chemical reactivity, I think, is one issue here of glutamine. They stick with the conformationally restricted glutamine in that way. And the interesting thing to us was now they had a different hook. Instead of an aldehyde, they put in a nitrile. And I won't show you the chemistry here, but nitriles will react reversibly with thiols, just like aldehydes do. So this is also a good bait with a hook on it to become a potential drug. In that article, they talked about the potency and effectiveness of this in inhibiting the virus, not only the enzyme, but the live virus. However, there were other issues with it in terms of bioavailability and some potency issues that led them to pursue a continuing SAR, again reported in that same article, which led to the first treatment for COVID post-infection drug treatment based on inhibition of MPRO. And here's the molecule. It's called nirmatrelvir. Sort of complicated now, but if you look here, you see they've maintained the conformationally restricted analog of glutamine. You see embedded the leucine residue, and they have a very, very different acyl group on here. And there's a whole discussion of that in the article as to why they came up with that. It didn't have really a lot to do with the inherent having the most potent inhibitor, but it had to do with bioavailability issues that they struggle with some of these other molecules. So that became the drug to treat COVID. If that molecule is called nirmatrelvir, it's in this box, and the drug is called Paxlovid. Perhaps you've heard of it. And Paxlovid, which received emergency approval in December 2021, if you take that drug within 5 days of having been diagnosed with COVID-19, you decrease the likelihood you'll end up in the hospital by, I believe it's on the order of 80 to 90%. Those are odds that I'm willing to take. In fact, ended up taking. But if you, so you get this package and then you open it up. And again, I mentioned, I actually came down with COVID on the 3rd lecture to Kyiv. At that day, I was feeling kind of not so well. But the next morning, I tested myself and I had COVID. And so I called my doctor. And since I'm in an age group that's more vulnerable, he immediately prescribed Paxlovid. I got Paxlovid. opened it up, and here's what I have to look at. 3 pills taken twice a day. 2 of these pills are nirmatrelvir, the active inhibitor of Mpro. The other pill is not active against Mpro at all. It is actually a molecule that was developed to treat AIDS that has as one of its effects the ability to inhibit critical enzymes, metabolic enzymes. And so what they found out initially with nirmatrelvir in their studies to see if it's going to be a drug and taking it through clinical trials is that this molecule doesn't survive very long in the body because it's quickly metabolized. And by putting in an additional molecule, ritonavir, and inhibiting the enzymes that metabolize nirmatrelvir, you can make it an active drug for treating COVID-19. So I take 3 of these pills, 1 in the morning and 1 at night. Not everybody can take these 3 pills because ritonavir has side effects of its own. In particular, I believe there are kidney issues that are associated with ritonavir. And so people who are going to be candidates for Paxlovid can't take this drug if they have kidney issues. So that just reminds us that, again, drug discovery is a pretty complicated process. You may have a good enzyme-based drug that's validated, good way of making it, but there are all these other issues that you got to have that go outside of just having something that simply inhibits the enzyme. So you got to be able to deal with these other issues at the same time that you don't screw up the enzyme inhibition. So that's where a lot of the work goes on. And in fact, in some ways, that's what we're doing in the Pseudomonas aeruginosa project. So that further catalyzed, we already were developing the lab when the Science article came out, but we were developing it with the idea of having the aldehyde hook. When the Pfizer work came out in Science a year ago with the nitrile hook, we realized our undergraduates could put that nitrile hook on too. So we developed an undergraduate COVID-19 Mpro enzyme inhibition project based on that. And our idea was, again, to look at the bait portion, but now in our combinatorial world, make as many different analogs of that bait. In one of our labs, we were going to keep as the same variable at the P1 position, the conformationally restrained glutamine, but we vary groups out here with the idea that maybe we can make another analog that will inhibit the enzyme, but maybe is metabolically more stable, okay, or it's more readily absorbed or meets some of these other characteristics. Also remember, and this is going to happen with Paxlovid, just like it happens with any other antimicrobial compound. When that molecule, when Paxlovid is out there long enough, we're going to see a mutant of Omicron or SARS-CoV-2 mutant that now has a mutated Mpro that no longer is inhibited to the same degree, or maybe there's some enzyme now that's going to chew it up even faster. So there's always room for improving or anticipating the need for other developments. This is classic in pharmaceutical chemistry. Converting enzyme area, captopril was the first molecule, but it's rarely used now because of many better molecules. In statins for cholesterol lowering, the early ones are rarely used. Converting enzyme, anyway, there's a lot of examples of that. So there's reason to continue this research. Nirmatrelvir is not the end of the day. So here's the lab synthesis that we developed with our students, our independent undergraduates. We make for them the conformationally restricted glutamine residue attached to polymer B, Merrifield resin actually. And they take that, they take off a protecting group, have the free amine, then they acylate with the first variable in this series, protected as a Bach derivative. Then after they take off the Bach protecting group, they acylate with a second residue. So we have just color coded the blue and the green variables to make this fully developed molecule, resin bound. And then they cleave the bond, the ester bond, to the Merrifield with ammonia. And by doing that, they simultaneously give us the desired molecules in solution, but they also now have a primary amide at the end. And you see all the possibilities we have here just from this simple thing where we can have hundreds of different combinatorial possibilities from the blue or green variables. Again, simple equipment. So this is all they need to conduct that synthesis in an undergraduate lab. And then, well, I don't show you the purification equipment. They use a simple kind of syringe-filled silica gel column to do the final purification, but that's easy to come by. I just want to mention something else because I have a very wonderful colleague in the Czech Republic, Viktor Kirknak. He's at Pulatsky University in Olomouc in the Czech Republic, not terribly far from Ukraine. And Victor is a very clever guy, and he's developed a whole series of simple equipment to do multiple, single or multiple solid phase reactions. So I just wanted to point out his equipment too. And I talked with Victor, and he said, if there's interest in Ukraine in doing some of this chemistry, he would be willing to supply one of these domino block synthesizers, which has multiple syringes that can use these resin-bound reagents for free to somebody who want to pursue that. So either be it a domino block, some of our colleagues use just a single syringe here to do their synthesis. So you can do it one at a time or multiple at a time. And of course, there's the billboard equipment here that also can do it. And by the way, I'd be willing to supply some of this also for free. There's no monetary interest in this. So with that synthesis in place, here is our grid for distributed synthesis that was generated. And you see here what we're doing. Well, one thing I should mention is that whenever we do an undergraduate lab or a graduate lab, we always insist that there be one molecule that every student makes that is the control molecule. That takes it into the realm of a traditional lab in the old days, to me, the old days where you make something that was already known, you showed that you knew how to make it, and that you did a good job of making it, and then you put it in the waste disposal can at the end of the day. Well, it still was valuable to have made it as a documentation of your mastery of the synthetic procedure. So we still have everybody make a control molecule. We had all our students make this control molecule, but at the same time, every student or student team will make an additional molecule as they're doing their work making the control molecule. And that additional molecule could be any one of these 30 other molecules. So those were our targets. We just completed this lab quite effectively, and I'll show you some of the results from that. So here is an example of just replicate. Again, remember, we're replicate people. So this is the control molecule. So everybody, every team, the team here consisted of 2 students working together. Team 1 and Team 31, I just picked out these particular LC traces to show you how pure the material was that Team 1 made and essentially replicated in terms of purity by Team 2. Every once in a while, we had how many teams? We had 60 teams. So every once in a while, somebody messes up, but that's the way it is, and they understand. So that's the control molecule. Here's an example, though, of one of the new molecules that in this case was made by Team 8. So Team 8 would have made the control molecule plus another new molecule. And Team 3 also made this new molecule. And again, you see LC traces of quite pure material. Well, let me compare for documentation that students can make molecules that are being by procedures that are pretty sophisticated and that are equivalent or even better than some that are being used in industry. So here's the solution phase synthesis published in that Science article talking about the discovery of nirmatrelvir. They start out with this molecule, they deep block it, attach the second amino acid, leucine, they deprotect it, they put on the final capping group. And this gave them a key intermediate. It's not the final, it's not nirmatrelvir, but it's a key intermediate in the SAR that led to nirmatrelvir, and all the work, the experimental, is in their supporting information. Let me compare that to what the students do in our undergraduate lab. They are given, again, this starting material here, but the key is, unlike Pfizer's work where they have a methyl ester, we have an ester too, but the ester is to a polymer bead. And that means all these steps that we go here can be done by simple filtering, washing workup steps. So we don't have to do recrystallizations. We don't have to do extractions. We don't have to do anything that you would normally have to do in solution phase. If you do this on solid phase, you can also do it on a small amount of material. So our students typically are given about 13 milligrams of this starting material, resin bound, and they carry out the sequence. Many students having never done synthesis before, they carry out the sequence and they end up after chromatography of the control molecule, approximately 50% yield, 12 milligrams. So how's this compared to what the folks in the science article did? Here's an NMR of material prepared by one of our students and typical of our students for the control molecule after they purified it by chromatography to get that 52% overall yield. That's the NMR. I know a lot of you will not be familiar with interpreting NMR, but a picture is worth 1,000 words. So I copied and pasted from the supporting information of the science article that led to the discovery of nirmatrelvir, this key intermediate that they had made, the same molecule that our students are making. And what you'll note is that, in fact, our material is purer than the material reported in the supporting information of the science article. Not a big deal. I mean, if you look at these are the three materials that you see that aren't present in the NMR of our students' material. But in fact, that's solvent, contaminating solvent. They just hadn't pulled off the solvent long enough before they took the chromatography. So their material was really quite pure, but it's just contaminated by solvent. But this is stuff that our students made. And this is stuff that could be done, I'm sure, in any lab in Ukraine if they had the equipment and had the students that have been well educated by you folks. So the final step in this process is going to be to attach the nitrile hook to the amide. And what you see here is the process that was used in nirmatrelvir. So they discover they take the primary amide, they treat it with POCl3, and that's converted into the nitrile. So we needed to show that we can do that with our molecules too. So we have an independent undergraduate student who took the control molecule and converted it into the nitrile. And again, this is a key part of the discovery of nirmatrelvir. If we had all the fancy spaghetti on this end, we could have made nirmatrelvir. So right now, we have an independent group of our students taking the molecules, those 30 odd molecules from the internal distributed synthesis of these primary amides, and they're taking them and converting them into the nitriles. At the same time, we are developing an MPro assay. We almost have it. We can do it again with a few students, but we haven't written up the procedure to the level of confidence that we say we can just present this written procedure to any student, and if they follow it, they'll be able to do the MPro assay. But we're close. I think within a couple of months we'll have that. So we'll have the coupled lab of distributed synthesis of nitriles and testing against MPro to come up with potential new drugs to replace nirmatrelvir when the time comes. So I'll summarize here by just showing you some of the schools that we plan to invite to this new COVID-19 drug discovery lab. We now have the confidence. We would never ask our colleagues to do this if we hadn't had this done by 6 different sections of students at IUPUI and reproducibly shown that the control works and that they can get these new molecules. But we're confident of that now. So we'll reapproach those who have been associated with our D3 dipeptide prodrug work, along with some of our colleagues who we haven't had involved in that project. And my hope is that when the time is appropriate, when you have the facilities open, when you can work without the constant disruption that's coming from this war, that we can help you if you are interested, be a participating, have participating schools in Ukraine. So in summary, here's the project, Distributed Drug Discovery. Our goal is to involve students in learning both synthesis and biology in the drug discovery process while they're working on these collaborative international projects for neglected and infectious diseases. We want to bring students together rather than killing each other. we want to bring them together across borders to show a common understanding and common goals. And again, I hope that we can involve you when the time is appropriate. Final slide, just acknowledging, as you can imagine, there are a lot of people to acknowledge, but I just kind of highlight here my colleague, Dr. Marty O'Donnell at IUPUI, and Gino Sammartoni. He's the guy who runs these laboratories. And if you can imagine on the scale that he's running them, it's pretty intense. If you were to run them, you just have one student or two students, so it wouldn't be as intense. But he's key. And then we've had these collaborators throughout our days developing this project. So thank you very much for your attention. And again, I hope this can be something of an encouragement to you to know that you're not alone and to see the invitation to be participants in this project. So thank you.
Thank you very much, Bill, for a very interesting lecture about medicinal chemistry and drug discovery distribution. And I hope we will find students in universities of Ukraine for your projects as well in the future, of course, and especially from Kharkiv National University. So, dear colleagues, do you have any questions to our lecturer about this very interesting topic? Please.
I have a question.
Professor Lipson, welcome.
Dear Professor Scott, could you explain, did researchers take in account structure of angiotensin converting enzyme inhibitors when they started this project?
No, I think so. You know, if I understand your question, you were wondering about the structure of angiotensin converting enzyme. Is that correct?
Yes, yes.
And which is very different in terms of its structure from the main protease. So no, I don't think they did.
No, yes. Thank you.
You're welcome.
Thank you very much. Another questions?
May I have question, please?
Dr. Harabet, please.
Hello. Thank you very much for a very interesting lecture. Actually working in the Kharkiv National University in medicine, in School of Medicine as a like given practice for the students, I was quite surprised that we had so many international students in our school. There was thousands of students, and the idea was to develop a department that would give also medicinal chemistry to this student. And I thought approximately the same, that students will do some chemistry that will provide a lot of compounds, and they will be also analyzed quickly biological activity. And you have realized this idea. It's incredible. That's why I'm a little bit excited. But the question is about fluorinated aniline derivative that was toxic, and you used like dipeptide from this derivative. This is initial part of your lecture, and you said dipeptide will be cleaved by the cell of bacteria, but when it will be cleaved, it will release again this toxic part of the molecule, the fluorinated alanine. And how do you realize this issue? How do you when this toxic molecule will be released?
So the hope would be, and this is yet to be demonstrated, but the hope would be that the toxic fluorinated phenylalanine would be only generated within the cell of the bacteria, and it would kill the bacteria. Now, the fluorinated phenylalanine will, you know, at that point will get out, I guess, if it's not actively metabolized, and could be toxic in its own right. But the relative concentration of that should be less than it would be, in the bacteria itself. And so we might not have to use as much of the dipeptide prodrug as we would of the parent fluorinated phenylalanine, both because the dipeptide will be actively transported into the cell and that it's uniquely made, regenerates the fluorinated phenylalanine within the cell. But this is speculation on my part, and this is the kind of thing that experiments will have to show us that yes, in fact, we do a good job. We do know these dipeptides, by the way, are good inhibitors of Pseudomonas aeruginosa. We haven't examined them systematically against mammalian cells, and we certainly haven't taken them further to do the appropriate, what a pharmaceutical company would be doing, the toxicology, to look at what effect these have in mammals.
Thank you very much.
Thank you. Other question, please, colleagues. Okay.
I'll just make one comment now, since one of your colleagues is interested in possibly doing this kind of a project within their own labs. We look at this as a tool that can be modified depending upon the needs and the objectives of a particular institution. So if somebody's really interested in digging deeper into the biology of the COVID virus, that can be a whole discussion within a class. And then a subpart of the class is involved in making these molecules. Or if the main objective of the particular class is peptide chemistry, they can focus much more talking about the details of the mechanisms of the reactions that are involved, and then use this project as an illustration of how that works. So I'm hoping people will realize that there are many ways that you can use this creatively in your own work outside the way that we're using it.
Okay. And I have questions about management of distributed drug discovery. The main question is intellectual properties and publications. How did you solve these problems in your work?
Yeah, well, that's a really key question. And I wouldn't say that we've necessarily solved them. In terms of publications, it's wonderful to be able to publish with all our collaborators on the publication and work together. It does involve a lot more coordination than I'm used to in terms of writing up a publication and working with other folks. There are some, I'll just open up we call it in the United States a can of worms. That's when you open up something and all of a sudden you got a whole lot of other problems. And the issue is intellectual property. I did come from a pharmaceutical industry and know well that sometimes patents are valuable in terms of providing the incentive for somebody to take something further on. So we love to be able to share information as much as possible. And that's part of our project. But someday we may have to deal with the fact that we want to make sure that if we're not the ones who are going to take this on to be a drug. It's going to have to be probably a pharmaceutical company or small startup company, something where they have an incentive to do it. And part of that incentive may be that they can get some profit from the molecule, and that may require having a patent. Mm-hmm. So we're trying to figure that out. And then I'll ask you the question, what happens when reproducibly that molecule has been made in Kiev and it's been made in Indianapolis by two separate students, and that was the first time the molecule was made? Who gets a patent?
Yes. And
I thought I got away from that by moving into academia. I thought, boy, this is wonderful. Finally, I can talk about what we're doing, share the information freely, and not have to worry about it being a problem. But
okay,
I'm going to ignore it for the time being.
Returning back to publications, did you include students to articles or not?
Yes. Yes, we did. So the initial concept, no, that was just me and Marty O'Donnell. The second paper was a collaboration with Lublin, Barcelona, Moscow, and us. And there were students included that And then we had a publication on, we called it a collaboration with COAD. That's a community for open antimicrobial drug discovery. But we had multiple schools involved, and we had many students involved because they were ones who were making the molecules, you know.
Yeah.
So they deserve to be on. And some of them not only were making the molecules, but they were developing these procedures that could be used in a regular lab.
Yes, and another question about financial support of your distributed network. Do you have a special grants, or where is your source of financial supporting?
Yeah, we've had sporadic support from different organizations. We had some early on some National Science Foundation support. We've had support when we went to Cuba from the American Chemical Society. They had a global innovation grant. We had support when we let's see, from COAD for developing this work. This is an interesting question. You know, I initially proposed this idea to the Gates Foundation back in 2003, and they were looking for ideas that they would then support other people carrying out with money from the Gates Foundation. And so if they selected your project, they wouldn't give you money, but they would say, this project we're going to support, and we're going to give anybody who wants to support this project money. So I submitted it to that, and it wasn't selected. There were like, I think 1,000 applicants, and they selected 8 or 10. So the odds were not very good. But when I reread my proposal, I said, this is going to be simple and inexpensive. And I said to myself, do I really believe that? And I do. And so a lot of this has actually been financed out of my own pocket. I contracted with a supplier to make that equipment out of my own pocket. If I believe in this, then I need to make sure it's cheap. And it has to be so cheap that a lot of this work can be financed by our department, by myself, by a few other benefactors. But, you know, it'd be nice now to have some larger group to coordinate this. And frankly, I'm getting older, and you know, I need somebody to be able to take it over and do, you know, the coordination. You imagine what Enamine needs to do to keep track of their virtual catalogs and all the samples that they make. And so we have, you know, tons of samples in our labs from our collaborators that have sent them to us for testing, but we can't do that forever. So this is a challenge.
Yeah, it was my third question about coordination. Okay,
Right.
Very nice. So, dear colleagues, do you have any other questions? I don't see. Okay, dear Bill, thank you very much for a very interesting lecture and very interesting realization of cheap and effective drug discovery process. International drug discovery process. And I hope Ukraine also in the future will connect with your project or maybe similar projects.
Very good. Well, thank you again for hosting this, Professor. It's wonderful that you're doing this.
So thank you very much, and have a nice day in the United States, and see you.
See you. Keep warm.
Okay, bye.